Okay, so again, just talk about it's citric acid cycle is not a closed loop, not just for glucose. And remember, I asked you to watch those videos, and they talked about all of the enzymes that are used to convert those molecules from one to another so they can progress throughout the cycle. Enzymes can be controlled. They don't just we talked about activation energy, and we talked about enzymes lower the activation energy and allow the reactions to occur more easily. That's the goal of an enzyme. They've got this active site that a substrate attaches to that active site and holds it in such a way to get it to more easily to a transition state so it can be broken down or put together. That's what enzymes do. Really cool protein molecules. So if they lower the reaction energy required for a reaction to occur, well, why doesn't that reaction just occur spontaneously or just occur all the time when an enzyme's present? It can actually. We actually have to sometimes slow down these processes in the presence of an enzyme because you don't want to generate too much ATP, for example. You generate too much ATP, we mentioned that is those molecules are actually not that stable. And if too many of them break up, that can emit a lot of heat enough to the point where it can actually kill the cell. It can basically bake the cell. So you don't want to just over generate ATP all the time. So there has to be some kind of mechanism in place to slow down the processes at various points to prevent buildup of some reactants or to prevent buildup of too much unstable compounds, whatever the case may be, or generate too high of a pH, too much of an acidic environment within the cell where other processes won't occur. If left unregulated, enzymes can have a very bad effect within the cell. And so how do we do that? Well, we've mentioned, I think a little bit earlier when we first introduced enzymes, we talked about competitive inhibition. So there are molecules that can attach to this active site, preventing other molecules from attaching to that enzyme. There's also what we call non-competitive inhibition, which normally takes the allosteric form. Here's a diagram out of your text. I think we've seen this before, but if not, you have what we call an allosteric site. This site, it's not the active site, not where the substrate attaches, but somewhere else in the molecule. That if you put an inhibitor in there, it changes the site, the active site, so the molecule can't be processed by the enzyme. Or some of them require the allosteric require an allosteric activator. They require some other molecule or compound to attach to the enzyme to open up the active site so the substrate can link in. So there's a couple of ways, right? So again, competitive is where something else attaches on the active site. Allosteric is where something else attaches in another location that changes the enzyme to where it can or cannot operate. And this is what we call a feedback loop, to where maybe it's not, it's not this product here that stops this first enzyme from working. Some other end product later on in the pathway can be used to prevent the first enzyme from working, and that slows the rest of this process down. Okay, so that can occur. And so once there's a low enough if there's too much concentration of whatever this block is, that's going to stop the process from ever beginning. But if once let's say this is ATP, for example, right? It's not, but let's say it is. Then if there's too much ATP in the cell, and some of those ATPs can bind to this enzyme and stop it from producing more of these pyruvate or whatever the product may be to prevent the cycle from going forward. It's not quite that simple, but it is. That's essentially what happens. It's just not ATP and pyruvate. This diagram is from your text, and let's see, can I? All right, we'll look back at this diagram here in just a second. All right, that's almost 5 minutes. I'm going to stop there.